Thank you, Rory. Thank you for the invitation. I'm at the Wellcome Trust Centre for Human Genetics in Oxford. We're interested in genetic susceptibility to infectious diseases, and I've been working in that area for over 30 years now. And as Sir Rory well knows, I'm getting to that stage in my career where often my main contribution can be to give a sort of historical perspective on the importance of the proceedings of the day. So very briefly, about 25 years ago, I was working in West Africa collecting samples for a case-controlled study of genetics, malarias that happened, and I spent a lot of my time persuading my then mentor, Sir Brian Greenwood, that 2,000 DNA samples wasn't a sort of excessive, self-indulgent number that we didn't really need. And, you know, it was barely enough to see anything in those days. These days, 25 years on, we work in international consortia, hopefully close to 20,000 TB samples soon, to try and see real, true, not false positive signals in looking for genetic factors in infectious disease. And that has made a big difference. We can see real signals now that are telling us about disease pathogenesis and how to impact on it, on disease. What we've never had the opportunity to do is to go a log further, not from tens of thousands to hundreds of thousands, to the sort of opportunity that Biobank provides. So from the genetic perspective, this is enormously important. You'll hear more about that from Hugh Watkins, I imagine, in the next presentation. But what Alex and I are here for is to talk about how exciting it would be to measure some infections in this cohort, not just from the genetic perspective, but arguably even more importantly, from the perspective of interaction between infectious pathogens and other diseases like cancer, cardiovascular disease, and a very important set of inflammatory diseases that you're all familiar with. So I'll leave it at that, hand over to Alex Mentzer, who's a clinical fellow in the research group in Oxford and has been leading on this very exciting project to tell you about the details. Alex.
Thank you very much, Adrian and Rory, for the introduction. So just under a year ago, I was part of a small group of researchers who was approached or in discussion. We were asked if we could think of a way to make this phenomenal resource of potential particular value to the infectious disease research community. After a small pause for thought and the consideration of the huge task potentially ahead, we had no hesitation in making a series of recommendations of how we could make this potentially feasible. Those of us who were involved in this are partly from Oxford, but also following extensive consultation from international experts in infectious disease. And we've developed a project proposal on the basis of this. We are largely motivated and inspired, I suppose we could say, by the biochemical biomarker panel, which is a fantastic initiative in itself. But that has really led us to where we are today. And as part of the project proposal, all those involved in it, quite an extensive number of investigators, all thought the most sensible and informative way forwards would be, again, to screen all 500,000 participants for evidence of prior exposure or carriage of a range of infectious pathogens that have suggested evidence for involvement in a range of chronic diseases. Using antibodies against these pathogens are tried and tested and validated, and probably the best mean that we have to test this exposure. Furthermore, there's good technological advancements now that means that we can do this for multiple pathogens simultaneously, and even at a size of, in a cohort the size of UK Biobank. And what's more, we further considered that, relevant to some of the points that have already been raised with the biochemical biomarker, it would be fantastic if we could look at individuals in at least 2 time points. Not only just to broaden the scope of research questions that we can look at or potentially answer as a scientific community, but also this would enable us to test the validity of the assay that we would use to measure these antibodies. So measuring these antibodies would allow us to look at various scientific questions, but there were 3 that were particularly on our mind when we were developing this proposal. Firstly, the antibody data alone. Looking at multiple pathogens simultaneously would essentially be the largest seroepidemiology study ever undertaken, especially when looking at multiple pathogens and multiple antigens within each of those pathogens. Then, obviously, combining that data with the data available, the eventual critical endpoints of UK Biobank, the events, chronic disease outcomes, we would be able to critically appraise and test the associations of those infectious diseases with chronic disease outcomes. But most importantly, as Adrian has alluded to, and certainly what I think is most interesting and most fascinating, although obviously I am slightly biased working in Adrian's group, is the potential to overlay genetic information so that we can really understand how host genetic variation can modulate susceptibility to the eventual chronic diseases, but also susceptibility to infectious diseases in general. Now, I'm sure there are two key questions that are on the majority of your minds. Firstly, what are the pathogens we're thinking of looking at? And similar to the biochemical biomarker project, this was no mean task and several hours of debate to come up with a list. But here it is as it stands. Hopefully you can read that. We wanted to focus on pathogens for which there was a significant amount of evidence for association with chronic disease. We also wanted to focus on those pathogens that we knew we could measure antibody against in a platform that could be readily transferable to the assay that we would use. Generally, these pathogens can be subdivided, as it were, into the herpes viruses, the chronic viral hepatitis, the HIV and HTLV1, polyoma and papilloma viruses, and a small selection of bacteria and parasites, which are known to induce chronic infection states which can be linked with chronic disease. And obviously, the selection of many of these pathogens was on the basis of the fact that many of these are linked with the development of cancer. And indeed, many of these pathogens are those listed on the International Agency of Research in Cancer Group 1 pathogen or agent list known to be carcinogenic in humans. But several, nearly all of these, have strong evidence for association with cancer, which hasn't meant that they've been translated into this highly carcinogenic class. But this raises two very important points about the project that we're proposing. Firstly, we would be able to reliably assess, confirm, or refute the association of these pathogens with these chronic diseases, as we would expect a large number of cases of various cancers to be attained through UK Biobank. And also, most importantly, or most interestingly to me, is the observation that even if individuals are infected or have been infected with some of these pathogens, how can we understand why some of those individuals don't get cancer? And with overlaying the genetic information, the other detailed phenotype information, I think would be able to make that more clear. Obviously, as Adrian alluded to, it's not just cancer. It's a variety of diseases, but most importantly, those which would be very well powered in UK Biobank to understand: cardiovascular disease, development of atherosclerosis, which infectious agents have often been linked, and those highlighted here in yellow are some of them. Again, the power to dissect that. Inflammatory disease, multiple sclerosis, autoimmune disease, again, pathogens far and wide have been associated with this. Are these associations real or not? Now, the second question, obviously, is how are we going to do this? As I said, there have been technological advancements. Again, hours of discussion. We think the most viable approach is to use a multiplex immunoassay platform, commonly marketed as Luminex. The benefits of this: it's sensitive, often far more sensitive than the ELISAs which are available, and I've got good experience of that with a number of these particular pathogens. Semi-quantitative, we can equate almost the outputs from the Luminex or immunoassay to a concentration of antibody in that particular specimen which you're measuring. And most importantly, you can multiplex. You can combine multiple reactions up to a 90 or 100 simultaneously in the same phase. What that means is we could essentially measure up to 90 antibodies simultaneously at the same time, same reaction. It's not easy. It's going to be technically challenging. We realize that. We've been working very closely with Dr. Michael Pavlito in Heidelberg, who I'm sure some of you may know. And he's been working with this particular platform for years. He has extensive experience. And most importantly, he's developed assays for nearly 80% of the antigens that we're interested in looking at for this particular panel. He's validated these assays against established gold standards other assay platforms to compare antibody concentrations against. But most importantly, he's had access to reference sera, which we know often through the gold standards are either positive or negative for those antibody levels. We want to take that one step further. We want to get independent suggestions of which gold standards to use and availability of reference sera. So for this project, we aim to work with a nominated expert in each of the pathogens so that they can provide us with relevant expertise and information. Once we already have those nominated experts for about 50% of the pathogens that we're working with, and we're already working closely with them during this project proposal development. But once we've confirmed the identity of those individuals, and once the project has been approved and we can go ahead, the first phase would be to develop and validate the remaining 20% of assays. This would largely be done in Dr. Pavlita's group, and then coincidental with that, we would be looking to transfer the technology from Heidelberg into the UK Biobank laboratories, as Tim has already been discussing, which is going to be crucial for ensuring quality control and consistency. This technology transfer, Dr. Pavlita has undertaken several times, both to developed and developing countries alike. Simultaneously to that, each of the assays would be validated independently, but of course we're going to be dealing with a multiplex reaction. We would ensure that the multiplex reactions would be validated in the same way each time as the individual assays, building up slowly with 5-plex, 10-plex, up to 50-plex potentially, and ensuring minimal loss of assay performance. There is the possibility, and we are open to the realization that some of the assays just won't work either on Luminex or in the multiplex format, and then we'd have to make a concerted decision as to whether we would take those assays forward in a different platform simultaneously, or whether we could think of another way, or whether they would have to be left out altogether. Once this assay, or probably 2 multiplex assays, I would imagine, were developed, then we would look towards a pilot study, 5,000 samples, both in Heidelberg and here in the UK, to ensure interlaboratory minimal variation, and also to ensure that the assay managed a relatively high throughput manner. Then we would step up to looking at 20,000 samples, 10,000 baseline, 10,000 follow-up from the same individuals, to ensure assay consistency inter-individual, obviously accounting for the fact of incidental infection in those 2 time points and loss of antibody persistence for some pathogens. And then finally, after data curation, we'd be looking to assay up to 600,000 samples by the time we would be at this point we envisage with the imaging follow-up samples and the other follow-up samples that Rory has mentioned. We'd be looking to assay 500,000 baseline and 100,000 samples as a follow-up at various time points. At the heart of this, as has been alluded to, is we have been determined to ensure that this is of high data quality that would be consistent with the rest of UK Biobank. We believe that working with the independent experts for each of the pathogens would make this viable and real. And importantly, having access to reference sera that we could use as internal quality control for each of the pathogens to minimize assay drift or batch effects. And importantly, we would use similar strategies as Naomi alluded to for the biochemical biomarker project to minimize systematic bias through sample selection strategies. We would expect when, if, hopefully, approval for this project goes ahead, we would expect the validation stages to take 24 months. We would expect the assays to be undertaken for all the samples within another 24 months, and therefore allowing for interim data analysis and quality control metrics. We would expect the whole project to be approximately 5 years. We believe that this proposal that we've set out so far is probably one of the only ways we can make this logistically feasible and economical, and to produce a data set of depth and breadth consistent with that of the rest of the UK Biobank data. There is, of course, the possibility of looking at more established assay platforms, such as ELISAs, for example, but we would have to look at a far smaller number of pathogens, or even antigens, really looking at a very small number of pathogens indeed. And the labor intensity of that would be far in excess of what we'd expect with the Luminex platform. and the costs would arguably be greater. We believe that this is, well, we hope that this appears to be scientifically robust, and we think it'll be of value not only to infectious disease researchers, but public health specialists, epidemiologists, geneticists, immunologists, just to name but a few. We are, of course, as is the theme throughout the day, we are, of course, very open to suggestions, comments, criticisms. We encourage anyone, if they want to come and help us, if they have any suggestions, we would be grateful for any advice. So please feel free to talk to myself, talk to Adrian, talk to any of the other UK Biobank members, or feel free to email us independently. We'd be happy to take any suggestions. Thank you very much.
So yeah, questions, comments, criticisms, whatever, to either Adrian or to Alex. So any questions? I can't see any hands. No. Do you have a sort of sense of the kind of cost of this sort of project on 500,000 people? I mean, obviously it's difficult to have huge detail in terms of all the quality control, but are you talking 50 million? Are you talking 500,000? Are you talking?
I think total costs in the initial preliminary costings that we've done is talking about £5 to £7 million, I think, for this, including quality control and development of the infrastructure to do this for all the pathogens that we've listed. Interestingly, it worked out, I think, at approximately £5 for the entire panel of pathogens, including assay development and validation for each sample that we would be measuring, which I think is vastly economical for this project.
Okay, I think you've taken people's breath away. There's a question. Yeah, there. So John Dinesh.
Yeah, thank you very much. Ambitious and exciting plans. Could you comment on both some of the pathogens that are proposed to be in and the extent to which serology might be informative, such as HPV, human papillomavirus, without the subtyping, or to what extent serology for Staph aureus is actually informative as opposed to a nose or throat swab? And then maybe also comment on what was considered but is not yet in the final shortlist and what was out, because I guess those are the kinds of in-out questions that would be relevant to hear about the thinking so far.
Of course. So to break that down, essentially we looked at as many pathogens as we could in the initial list, and we thought about which would antibodies be most relevant for, because we always had in our mind that we'd be working with antibodies, and which would just not be feasible. And that immediately cut down several, and then combined with that, as I mentioned, those with significant amount of evidence for association with chronic disease. There have been many, so syphilis, for example, treponema, which obviously fell out because of assay validity, multiple pathogens that we've looked at. So a number of bacteria, which we looked at, which just wouldn't be relevant to chronic disease outcomes, which are all excluded. The list is vast. In terms of particular pathogens, you're quite right in terms of which antibodies are informative for. So I didn't detail the subtypes of human papillomavirus that we'd be looking at, but we would be mainly looking at the ones most strongly associated with cervical cancer, 16 and 18. There's still debate as to whether we'd look at other subtypes, but I think we're just going to be looking at 16 and 18 at the moment. We think that'd be most informative, and no one, we don't think, would have been vaccinated against those in the group that we'll be looking at. So we expect the titers for those antibodies to be generally very small, and we would be prepared for that. Obviously, Michael Pavlita has a lot of experience in working with those. And in terms of Staph aureus, that is a very, very good point, and we think that's particularly exciting. We've actually got a collaborator, David Wiley, in Oxford, who is in the final stages of validating antibodies that are very good markers of carriage of Staph aureus and being able to differentiate carriage versus acute infection. And if that assay is validated on an ELISA platform, which he says is within 12 months, then we would be very excited to take that forward and look at the entire UK biobank, because it just hasn't been looked at before.
Great. Thanks very much.